again. Good evening, good morning, good afternoon, every one of us over there. My name remains Olatunde Michael, and today I've come again talking about TB genomics. TB genomics, and today our focus is going to be on what is TB genomics. Our focus is going to be on pangenome. What do we mean by genome? All this we are going to actually see what it means and what could be the application of pangenomes or pangenomic analysis in the research, Mycobacterium tuberculosis research, and what are the implication and interpretation of what pangenomes is. Don't forget that this is also a continuation of from wet lab to the dry lab, looking at the complete workflow of what is going to take if we are trying to get immunogenic genes from multi-drug resistant Mycobacterium tuberculosis, which could we are looking at the potentiality of making use of it in vaccine development. And just like I said, this work is a complete work that was actually conceived by a researcher. And just like I said, this is a quick general workflow or what is called the standard operating procedure. So this involves the following activities: pre-sampling activities, where we talk about identification of stakeholders, advocacy and consultation of, with stakeholders, preparation of research instruments such as consent forms, recruitment forms, and a lot of time was spent on ethical approval. Then apart from that, there was a time that there was a recruitment of field workers from each of the study sites. Initially, what was actually conceived was to carry out this research in 5 out of the 6 southwestern states in Nigeria. Initially, the name included Oyo State, Ondo State, Ekiti, Kwara, Ekiti, Lagos, and Ogun States. However, due to some bottleneck which has to do with non-approval of the ethical approval of time, and as well as and considering some other factors, we are able to actually focus this research on in Oyo, Ondo, Ekiti, and Kwara State. Part of what actually was actually responsible for inclusion of a state like Kwara State is that the data from that side is going to be a kind of a representation as a kind of Kwara State was supposed to serve as a gateway to the southwest. Okay, so after that collection of samples were done, all these were transported into the laboratory where the contamination of those samples took place. The use of a gene expert to be able to classify this Mycobacterium tuberculosis into multi-drug resistance, sensitive bacteria, as well as and so on. Then smear microscopy was carried out using AFB staining technique, that is acid fast bacilli techniques. That you should know that Mycobacterium tuberculosis is quite different from any other gram-negative or gram-positive bacteria because of the possession of mycolic acid, apart from the peptidoglycan layer that we have in gram bacteria. So Mycobacterium tuberculosis is notoriously complex because it has mycolic acid, moramic acid, and moraine, and so on and so forth, in addition to this, which will actually require to be dislodged even before the cell wall will now be stayed to all these common dye. After that, we look at isolation of this sample was done using low stringency culture media. So a lot of that was actually scheduled to have been presented earlier on in this as a kind of a progress report too for this work. So this also included molecular analysis, DNA extraction, PCR, library preparation, and whole genome sequencing, which was actually carried out using um, Illumina. So after that, we look at quality control using Galaxy, FASTA, Q, and so on and so forth. Where we are going to focus today is just like I said, is pangenome. What do we mean by pangenome? That's going to lead us to a different aspect of this. And don't forget that I told you we actually started with installation of Panaroo, okay, where we install Panaroo, we install Ubuntu, we install Anaconda. All these were done in the installation were done in the Anaconda terminal. So whereby we entered all these prompts until we are able to successfully install it before we actually stop. So and just like I said that all of you should, what my students in this cohort group should make sure that they update their status up to the status whereby they are expected to continue from that of today, that is getting the installation successfully done. That is the installation of Panaroo, the TB profiler, uh, Prokka, and the likes. So today we are, just like I said, we are going to actually focus on the Panaroo, okay? And this time around is going to be um, genome analysis, okay? Now let us go on to what, because somebody might just be asking us, why are we interested in okay, why are we interested in genomic analysis? So what I did was I wanted to know, and I now prompted ChatGPT by attaching the molecular workflow, that is the one we have been using before now. I attached it and I asked ChatGPT to help me to generate content that I can actually use for this kind of lecture. And this is what um, it has given me. That is a ChatGPT was able to provide all this for me. And after that, um, after the um, that has been done, so ChatGPT proposed that this kind of lecture should proceed in under 12, I think 12 chapters. The first one is a what is a pangenomics, why Mycobacterium tuberculosis need a pangenomics in terms of the strain diversity is needed, virulence variation, antimicrobial resistance difference, and vaccine design. What will be the implication of all these pangenes in the area of vaccine design? So just don't forget that um, now let us now look at the general workflow when it comes to pangenomic workflow for Mycobacterium tuberculosis. Now, the goal, the goal of this presentation is that we want to use Panaroo to build a pangenome of Mycobacterium tuberculosis, so that we can, number one, we should be able to see the core genes which are present in all strains. So all these one are good for vaccine targets. So the accessory genes, these are present in some genes, and these are important. They are very useful for antimicrobial resistance, as well as when we are interested in virulence genes. Then those ones that are unique, that is, they are only present in only some lineages. So this serve as a possible biomarkers for lineage. That is, anywhere you see all these unique genes, they are going to be very useful diagnostic tool to diagnose the outbreak, resistance, and so on and so forth. Now, just like ever before, if you want to design a vaccine design, that is in silico vaccine design, what you do is you subject your raw sequence, which was in form of FASTA So you subject it to um, the galaxy, that is under galaxy. We are able to make use of either we are able to make use of the fast QC. So thereafter, there was also another quality check and trimming of adapters. I've explained all this for us earlier on. So the genome assembly was done with what? Genome assembly was performed by speech, while the genome annotation was performed by Prokka. Then the level at which we are is to run Panaroo on all GFF files. We are going to see all this when we go on. So after that, the interpretation of our data will now lead us into those ones that we can make use of for vaccine design. Those candidates that are drug discovery candidates, we are going to see them. Then those ones that are of antimicrobial resistance studies, we will see them. So, very simple theory. That is, if somebody is asking you, what do we mean by pangenome? What are the relevance of pangenome in research involving Mycobacterium, tuberculosis, and some other? So, number one, genome. Genome is, if you come across the word genome, this is talking about all genes in one strain, all genes just in one strain. But we are talking about pangenome. We are talking of all genes seen across many strains. So, and these are divided into core genomes. These are genes that are present in all the strains. Accessory genome, these are genes that are present in some but not all strains. So this one actually differentiates or distinguishes one strain from another. Core from all, present in all. Then we now have unique because it is found only in one strain. So we are going to see all this when we go on. But for Mycobacterium tuberculosis, it is very useful for us, MTB research, because some lineages carry extra genes that improve survival in the host. Don't forget about different mechanisms that Mycobacterium use to survive in the host. So some of these genes are used for survival in the host. Then some of these genes are lost or mutated in drug-resistant strains. So all these are very important. Then we can also find very stable, conserved antigen that are vaccine candidates. So because we are going to focus on the core genes in designing this. So what do we mean by Panaroo? This is a graph-based pangenomic tool that, number one, takes annotated genome in form of GFF and FASTA, corrects the misannotation and frame sheet. Then it's going to cluster genes if annotation is messy. Then the fifth one is that it's going to give us clean presence or absence of a table and call gene alignment. Okay, so the input that is, we want to work on this. What are those things that you're supposed to have on ground before you be able to begin working on a Panaroo, which has to do with okay, so which has to do with diffusion of this. Okay, now the setting up environment. So if we are in um Anaconda platform, terminal, so you just be inputting all this code, you just be inputting it, and when you are inputting them, enter them, make sure you also hit your button which has to do with enter, and you have to take notes, take care, be patient until the system is giving you command by command, okay, step that you need to grow if you are working, if you are very serious working on this. So, and this also has a lot of, let me say, two or three environments. So the first one is Ubuntu. All these are installed along this Panaroo. Okay. So, having said that, you see, don't forget what we said. Okay, what is Panaroo? Okay, we have done that. We have done that. So, um, so we've done that, and we got to the level which has to do with annotation. We do annotation with Prokka. So, and when you want to do that, you want to do it in Anaconda terminal. Just come and copy this code, and you go and install it there. Whenever you are successful installing it, the system is going to let you know that we need to run this. Okay. Now, without wasting our time, don't forget that today we are focusing on pangenome, pangenome or pangenomic analysis, analysis of Mycobacterium tuberculosis. Okay, so let us just. I want you to see this illustration. What we mean by pangenomes means that don't forget that this is a set. Pangenomes are set of genes in more than one strain. But if you are talking of genomes, we are referring to just one strain, the totality of the genes in that strain is what is called genomes. Whereas, okay, so when you look at this, you will see that this green color, all of them, all the three, strain A to C, they have it. That is why they are called what? The core. Now, thereafter that, we also look at another types, another another set of genes that you can also find when you are running or when you run pangenomic analysis. So the second one is going to be accessory, whereas the third one is going to be unique. So in unique, when you look at this one, you see that it is only unique to strain C. Whenever you look at this color, you will see that is found in two. Okay, this one and this one. So all this one are called the unique genes. This one's the accessory, okay, because it's only found within, maybe between two out of these three, you find it there. But when you talk of core is really a core because it is the intersection. If you draw the three sets, I want you to represent. I want you to represent this in. Okay. Let me see. Let me see if ChatGPT can do that. I want you to represent pangenome genome in the example above with Venn diagrams using the above illustration. Okay, let us see what ChatGPT is actually telling us. But what you should know, even from there, is that okay, Okay. For some of us that are conversant with sets, we will know what we actually mean by that. Okay. So let this one, let ChatGPT continue that. Generate the Venn diagram. Okay. I want you to generate it now. Generate the Venn diagram. So you enter the prompt and you click on generate image. So let us see how this one will be generated, because that one is going to be more appealing to our eyes and is going to provide more explanation, a friendly, a beginner's friendly explanation about what you have said. But looking at this, let us just look at the illustration now. Okay, genome of hypothetical Mycobacterium tuberculosis strains using Venn diagram illustration. Now, let's say we have 4 different strains of Mycobacterium tuberculosis, and all these strains are this A, B, C, D. A, B, C, D. Now, in gene 1 is green, gene 2 is this. This is gene 3, this is gene 4, gene 5, gene 6, gene 7. So if you look at this green, if you look at G1, G2 to G3, G1, G2, G3, they are found in all. So that means gene 1, gene 2, gene 3, they are what? They are core genome. Okay, you can see it. Core genome shared by all the strains which appear in overlapping region of this. So intersection B, intersection C, intersection D. This is that. So because it is found in all. Now looking at the accessory genes, these are shared by some, just some, but are not all. So looking at it now, you will see that like this one, G4 is found in A, found in B, and found in D, not found in C. So G5 found in B, C, D, but not found in A. G6 found in only C and D, but this one, so you cannot see that G4 to G6, these are called the accessory genes, because they are only found in some, they are called the accessory gene. Whereas G7, because it is only found in species D, like strain C, D, that is telling us that it is unique. It is unique. Now, if you now want to design vaccine, antibiotic resistance, and you want to, so which one are we going to focus on? So that means for vaccine design, we are going to focus on the core ones, those ones that are what found in all. So you can see it. Okay, you can see it. Now test-based Venn diagram. So you can see core found in G1, G2, G3. So MTB A, B, C, D. So that is a accessory. This is accessory because it has an overlap. Okay, let's look at it. So this one can explain it better. Fourth Venn diagram, you can see 23. This is a core. This is A, B, C, D. So you can see this 23 genes combine all the 4. 25 combine only B and C. Okay, and 7 is only unique. So we need to know all this so that we will be able to know which one and which one are actually very useful for you whenever you want to. So now looking at this is another example. Understanding pangenome using hypothetical MTB strings A, B, C, D. A is drug susceptible, that is, it is sensitive. B, it is resistant to INH. Okay. C, resistant to this. Then Mycobacterium tuberculosis, which is D, they are resistant to only uh, two. Are resistant to two drugs. That is why they call it multi-drug. So when you now look at this, each strain has set of genes. So and let us have them in this color. So gene 1, that's all of them, because uh, all of them will have they must synthesize their cell wall. So the genes, all those genes that will be responsible for synthesis of cell wall are going to be core. And gene 2, for example, those ones that we call them housekeeping enzyme, they are called. So if you want to know more about housekeeping enzyme, let us see what ChatGPT will still tell us about housekeeping genes. So let me try and run another prompt, okay, for ChatGPT. So what are my question is, what are housekeeping genes can be found in Mycobacterium tuberculosis? Are they found there? Can they be used for drug design, design, vaccine development, and AMR surveillance? Okay, so what are they? Let us see what ChatGPT will tell us. Okay, housekeeping genes are the essential life support genes. Essential life support genes like oxygen for of any cell. They perform core functions needed for basic survival, such as, so another word for housekeeping genes are essential. For example, energy production, DNA replication, transcription and translation, cell wall maintenance, metabolism, and so on. So because they do the job the organism cannot live without, they are highly conserved. That means they don't change much across the strains. So what you find in strain A, you find them also in strain B, you find them in C, you also find them in D. So then stably expressed at all times. You know that they are consistent in. So don't forget the law of Charles Darwin, Jean Lamarck that I told you about, which is about theory of use and disuse. The organ or the gene that you tend to use from time to time, you will develop it. And the one that you do not need for survival, you tend to disuse it. So that is that. They are rarely mutated because they are very, very stable. So think of them, the housekeeping, they are like the engine part of a car. If one fails, the car will stop. So are they found in Mycobacterium tuberculosis? Yes. Mycobacterium tuberculosis has many housekeeping genes. For example, the one they use for DNA replication are all these: gyrase, transcription, this set of genes. Translation for protein synthesis, you need them. For metabolism like Krebs cycle and so on, you need all these genes. And cell wall, cell wall, you have to talk about muramic acid A, muramic acid B, and all this one. So even the famous drug resistance markers, which are common, which come from essential pathway. So can housekeeping gene be used for drug design? Yes, but with condition. Because housekeeping genes encode essential pathways. Inhibiting them can kill the bacterium which makes them good drug targets. Okay, so these are the set of genes that when you found them in Mycobacterium tuberculosis, they are they can be targets for drug design. Okay, and don't forget the classes and classification of drugs or antibiotics. So you can see, can they be used for vaccine development? Yes, but not the first choice. They are essential, they are conserved, they are expressed inside host cell, and they are often immunogenic. So these are the conditions that you must know whenever you are walking through your journey in vaccine design, antimicrobial resistance surveillance, as well as as well as a vaccine design against the immunogenic bond. So that is a pangenome. Pangenome means that you actually need to after the annotation, so you should be able to know those genes that are common among the strains, that we call them the core, and those ones that are common among only some of the strain or species, these ones are called the accessory. Whereas those ones that are found in only one strain, they are only one strain, they are called the unique genes. So they are less exposed on the bacterial surface. So they don't usually produce strong antibody. Uh, so most successful TB vaccine targets. So we target any of these. Uh, can housekeeping genes be used for AMR surveillance? That is antimicrobial resistance surveillance. Yes, and they already are. Housekeeping genes are some of the most powerful markers for AMR, that is antimicrobial resistance detection in TB. Why? Because drug resistant mutations frequently occur in essential genes. And you can see all these genes and their implication. So if you see RPOB, so it is meant for rifampicin, and the mutation that occurs in that place is so after that, you can see these are part of the mutational changes or mutational resistance gene that you can see. Okay, so all these can be classified by TB profiler microbe, WHO mutation catalog, and whole genome sequence-based DST pipeline, that is drug sensitivity testing. So you can now see that summary for you is that anyone that you want to use for drug design, they are housekeeping genes very, very suitable. So that is why you have yes here. So many TB drug targets essential stroke uh, housekeeping pathway. So vaccine development, vaccine development possible but not ideal for vaccine development. Low surface exposure and better for T-cell vaccine. So these are some of the distinction that you need to make by the time you are running your this thing pangenome. So don't forget that I have given you a lot of information about why pangenome is actually needed. And don't forget that also in this exercise, the workflow so far, I told you that we are making use of de novo assembly and annotation. So we are not making use of a reference. We are not comparing our unique genes or the sequence we have. We are not comparing it with any reference, because sometimes comparing it with a reference are often being biased, and sometimes we have many uncleared that okay. Okay, these are exactly what you say. So what we are saying now is just that Panaroo, okay, we are going to we are going to make use of this to explain what is called. Hmm. So this has actually divided the cohort group into three. Just like we have it, fasting design group. They will use core genes because they are present in all genes and are stable antigen. They are known to be stable antigen. So for drug design group, what you need is what you should be looking for are essential core genes, conserved metabolic genes, that is enzymes, unique structure proteins, consistent drug binding pockets, and those of you that are in the third group, that is the antimicrobial resistance, your focus should be G5. This is the drug resistant gene, which are present in MTB, B, C, and D. So, accessory genes explain why resistance differ among uh, strains. Okay, so this is a how can you annotate? How can we annotate genome and identify after we must have used Panaroo? Okay, so it takes your annotated genome and automatically. So after you must have run your Prokka. So the file you have from Prokka is called, I think it's called GFF. So it is that one you are going to upload, or you are going to um, input into Panaroo. So which genes are shared, that is, which are core genes, okay, which are variable, accessory. Using Panaroo, you'll be able to access it. Which one are unique to this string, you will also do what? Access it. So um, without saying much of our time once again, this is another opportunity to learn bioinformatics tools that are used for Mycobacterium tuberculosis research, especially this time. So I want to really thank you for your listening attention. And in my subsequent videos, we are going to also lay emphasis are going to lay more emphasis on this. But you can see that I've been mentioning, coming across what is a pangenome, so that you'll be able to understand what pangenomes are, the classes or classification of some genes as they are present or inherent in Mycobacterium tuberculosis, especially those ones that you can use in designing vaccine.